All right, so our last video here is for this unit is cell cycle and DNA replication. So with the cell cycle, I'm not going to cover too in depth because y'all do it in lab. So with this, cell cycle starts with interphase. This is the phase before we actually start dividing. It's just we're preparing, we're getting ready to. So within the interphase itself, we have three phases. We have G1, the S phase, and G2. So within G1 phase, this is just when your cell is growing. Your cell's growing like normal, producing its organelles, nothing big's happening. Now in the S phase, so again, remember an interphase itself, we're preparing for division. So within the S phase, this is where we start to replicate our DNA because we need a whole nother copy of DNA when we divide the cell so that one set goes to one cell, the other set goes to the other cell. So this is what she refers to as DNA replication. So with DNA replication, there's several steps here, and we don't really go too in depth with it. It's just these basic steps. So you just kind of know the gist of it. First things first, you unwind the DNA, and we unwind the DNA and break those hydrogen bonds in the middle because the hydrogen bonds will hold the two strands together. Think about like a ladder. You take a ladder, if you were cut the pegs in the middle, you'd be separating each side of the ladder. That's what you're doing to DNA. So with that, we're going to be using DNA polymerase. Remember in transcription, we had RNA polymerase because we were making a copy in mRNA. Well, with DNA replication, we want to make an exact copy of that DNA. We want to do another DNA strand. So we need DNA polymerase. We don't want RNA polymerase. We want DNA polymerase with DNA replication. So within that, you don't need to know too much about the leading and lagging strand. You just need to know that that leading strand is copied in a continuous motion and the lagging strand is copied in segments. So think about your lagging strand, think about lagging in video games or lagging in general. It's very choppy, so broken into segments. And then within your notes, you can kind of read about the 3' and 5', how we read which way from 3 to 5, 5 to 3, what is it? So you can kind of read about the 3 to 5s. And then once we're done copying the DNA, we rewind it back into a double helix. And that's the end of the S phase and DNA replication as a whole. Then we enter G2 phase, which is the centriole replication. Is we finished that. We're done making the centrioles. Now we're actually ready to divide, and that's when we get into mitosis, which you talked about in lab. So these are your mitotic phases. So first one is your prophase. So prophase, this is when we have the DNA in chromosome form. We start to break down that nuclear envelope. The centrioles move to the opposite ends. You can see that in the picture below. So we're starting to break down. We have our chromosomes are made into chromosomes. We're starting to break down the nuclear envelope, and then our centrioles start moving to the other side. Then we get to metaphase. So metaphase is over here, and this is when our chromosomes are all lined in the middle, and our centrioles are on the opposite side, and then our chromosomes are all lined up down the middle. Anaphase, we start to split. So the sister chromatids, which are basically each part of the chromosome, are split apart. So they're split apart into single chromosomes. That's anaphase. Now telophase is when we actually start having the chromosomes move to either end and then recoil into chromatids. And that's when a new nucleus, a nuclear envelope form. Now, if you notice they group telophase and cytokinesis right in with the same picture, that's because they're happening basically simultaneously. With telophase, cytokinesis is occurring basically right after, if not during. So this is when we start dividing the cytoplasm and we actually cleave or make the cleavage between the two. So we cut between the two and separate into two daughter cells. And that's the end of mitosis.